No, I haven't, but I have colleagues been there and also from the university and from the university. I have colleagues there in Brest, and it's perfect. So I think it's the So do you have there's not related to any university, right? No. It depends on the main city. Which makes it kind of interesting that the people consider a little bit of tension there. Yes, we can. Okay.
Okay. So, ¿podemos empezar entonces? Okay. Okay, good morning everyone. Welcome to Gran Canaria, to the University of Las Palmas de Gran Canaria, where we have the venue for this workshop on the European Marine Biological Resource Center. And we will have these 2 days workshop just to discuss and to present the results that we have already got from the project. And it's very interesting because I and we will present us some of the objectives of the project and the meeting, but it's very interesting because we have got an extension from the project, and it has been possible that we join the EBB project. So here at the university we are very happy to this. Okay, first of all, I will introduce the Director of Innovation and Transference from the university, from the Vicerrectorado of Research and Transference of the university, which is Professor Sebastián López, who will give you the welcome to the session.
Thank you very much, Juan Luis. So good morning, everyone. So on behalf of the Vice Rector for Research and Technology Transfer of the University of Las Palmas de Gran Canaria, who cannot be here today, it is my pleasure to welcome you to this European Marine Biological Resource Center workshop. As you perfectly know, much better than me, this European Marine Biological Resource Center is a European research infrastructure that provides researchers and companies to access to marine organisms and the facility to study them, including experimental facilities and technological platforms. So having said this, I see no better place than this, that the University of Las Palmas de Gran Canaria and Gran Canaria to celebrate this workshop, since our university, I don't know if you are aware of that, but has longly recognized the vital role that the marine environment play not only in our local ecosystem, but in an economy and our collective future. So the blue economy, which includes many disciplines like marine tourism, renewable energies, fisheries, and biotechnologies, among others, has always been in our NDA, has always been a strength of our university. For example, back in 1989, we created the first faculty of marine sciences in Spain, which is significant for our university. And moreover, different research groups and different research institutes are devoted also to the blue economy sector here at the University of Las Palmas de Gran Canaria. So a good example of the excellence achieved in this discipline in the blue economy is in fact the Spanish Bank of Alguil, which is the organizer of this workshop, which is a leading actor in this field, thanks to among others, to the excellent work made by their scientific directors, Professor Antera Martel and Professor Juan Luis Gómez Pinchetti, that I believe that it is fair to recognize and to acknowledge here. So our university has an an equipped workforce to address the unique challenge and harness the immense potential of the blue economy. This in turn, which is our vision, can drive innovation, can create jobs, and position our university, the University of Las Palmas de Gran Canaria, as a hub, as a technological hub for ocean-related industry and initiatives, which, as I said before, is our ultimate goal. I am sure that this workshop will definitely contribute to achieve this ambitious objective, so I would like to finish by wishing you a fruitful workshop as well as a pleasant stay here in Gran Canaria. Thank you very much.
Okay, yeah, thank you, Sebastian, for the words and the welcome from the university. Yeah, well, just to introduce ourselves for those of you that has not met us before. I will introduce my colleague Antena Martel, which is the scientific director of the Spanish Bank of Algae and the curator of our collection, which is a very tough work. And also we have Ignacio González Bonilla, which is our manager nowadays. He manages all well, he's very helpful for the management of the projects we have and the project we apply. So he has been organizing the whole meeting, which is very good for me also. And I myself, I'm Juan Luis Gomez Pinchetti. Well, I have met many of you before, and well, I'm also working at the Spanish Bank of Algae at the university. Okay, because I think this is a very small meeting, and we have met each other probably before. I have a very short presentation just to introduce you who we are, what we do, and how we are going to proceed these 2 days. I will move to the. Wow. Okay. So as I told you before, we are part of the University of Las Palmas de Gran Canaria. We are nowadays in this building, which is is defined as the foundation of scientific part of the University of Las Palmas, which play a very important role in the research and the innovation and the transference for the university. And for example, they help us with the management of our laboratory and our projects. So we share here buildings and capacities for the university and the Scientific Park Foundation. We as Spanish Bank of Algae are part of this structure, and you see this is the main university campus for the university, but in a place called Taliarte, where we will move tomorrow, we have what we call the Scientific Marine Park of the University of Las Palmas de Gran Canaria. Here in Taliarte, we will visit the Spanish Bank of Algae, but we will have around other infrastructures we are related with the aquaculture science, which is very important for the university. We have the institute called EcoAqua, and we have also the Institute for Oceanography and Global Change, which is also there, where is a very big infrastructure, and different groups of research are doing the work in this area over here. We have different laboratories, and we are joined also to what we call the PLOCAN, which is the Canarian platform, the oceanographic platform of the Canary Islands, which is the building that you have seen in the middle of the sea, just coming from the airport to Las Palmas City. So there is a big potential here for marine research. We are all related, very close related with the Faculty of Marine Science, and the potential and the work that we have been developing in marine science in the Canary Islands is very strong and very important nowadays in all this field of research. We, as part of this marine scientific campus, we are here in the harbor, and this is a small facility that we built up starting on 1989, so quite a long time ago, and we have been working here just focus on the scientific or research and development in the field of algae biotechnology. That means that we have been working both in cultivation and also in characterization of the biomass and the possibilities for innovation and development of the algae sector in the Canary Islands. Well, within the Spanish Bank of Algae, we hold a very important collection, which at least we think that is very important, that we have been working with in the last 25 years. Nowadays, we hold more than 2,000 strains of microalgae and cyanobacteria, and in the last years, because of our background was related also with seaweeds, we are just, with the experience we have also with the collection, we are introducing seaweeds in our collection, so the work is quite hard, and we will show you tomorrow all the facilities and the work we do. Yeah, during these 25 years, we have obtained different certifications, which are very interesting, and I think some of them can be an example for the work we are doing in EMBRC and the project, the EBB project. As far as they confer to us the possibilities to work, just not only in research, just also giving services and relationships with other collection, just not only from the marine environment, just also with the microorganisms in Europe. So the possibilities are very high, very high, and we try to go ahead with all the possibilities that we got from the collection. Yeah, together with the work in the collection, in the culture collection we develop, well, we have some mission and vision related to the field of the algae biotechnology and the importance that it can play in the Canary Islands. So we develop different strategic line or line of research which are related with the conservation of biodiversity, very shortly, conservation of biodiversity and the characterization of this biodiversity, which is, well, sometimes has been not very much studied in the Canary Island. I mean, when we start with the microalgae collection, the reference and the information we have got from other studies was almost nothing from the Canary Island. So we have developed technologies and protocols for identification of algae, and with our background, we are very much interested to go ahead with cultivation possibilities and the utilization and the development of application for the biomass that we obtain from our culture systems. And in the last year, we have been on a strong work on the possibilities for obtaining bioactives and the development of application around the potential that we got from algae in general. Well, we work in a very clear line that most of us we work on that. We go from the bioprospection to the biomass or to the biodiversity and genetic material conservation, and from this point we go ahead with the production of biomass both at laboratory and pilot plant scale. And from there, we make all the characterization of the biomass, and we see what possibilities we got from this information that we get from the biomass we produce. This is very much aligned with the strategy of EMBRC that you can see here, very simple, which is what we call here discovery pipelines. Okay, I must say that we come into the EMBRC infrastructure just last year, and through the Spanish node of EMBRC, and we are trying to go ahead and to take you at the same time in the infrastructure and see what possibilities we have in the world that we can develop with all of you. So we work in these basic 5 points activities, from the microalgae isolation and the growth optimization or characterization, and the possibility to develop products and services just coming from algal biomass. This is very much on, I insist, is very much aligned with the strategy of EMBRC and the relationships with other European infrastructures, which are, for example, AquaExcel, which is very much related to aquaculture. This is important to say. Our university is also part of AquaExcel through the EcoAqua Institute of Research. And we are nowadays in EMBRC, but also we are part of MIRI, so which is very much related to the possibility to develop technology and research and on the use of microorganisms, and it's very much related with this strategy of the European infrastructures, and particularly the center one, which is EMBRC. Okay. Some important points. We are doing a very nice job in relation with the possibilities of this sector in the Macaronesian region. We are part of a Interreg MAC project, which is called Rebecca, together with people in Azores and Madeira and Green Cape, and also some parts of, some universities in Mauritania, which is what we call the Macaronesian region. And we are looking for the potential of microalgae and algae in general in this region to try to develop the sector, the biotechnological, algal biotechnological sector in the region, which is, I think, very interesting, together with the work we are doing around the Atlantic area. And another important point that we will also discuss today, here in Gran Canaria, we are part of a platform which is called Bioassist Gran Canaria, which is, is a platform formed by the local government of Gran Canaria, which is called the Cabildo, through the Society for the Economical Promotion of Gran Canaria, the ITC, which is a public company from the Canarian government, and the university through the EcoAqua Institute and the Spanish Bank of Algae, just trying to develop the sector, the industrial sector of the algae in the Canary Islands, and particularly here in Gran Canaria. We have been working on that for 5 years now, and we have been finding what are the bottlenecks and the regulatories and the environmental issues related with the activity that we are promoting. But we are going step by step, trying to go ahead with this possibility. And in the last year, we have we have worked in the preparation or the application for the incubator of Gran Canaria for the sector of algae. These are some of the photos you see here at the center of the ITC in Pozo Izquierdo, going to the south of the island, where we are developing an incubator for companies to join us and to start the development of the activity under a collaborative area, which holds just not only the help on the development for application for permissions and all the technical stuff, just also to help them through the scientific knowledge that we have here in the Canary Islands, together with the people at the ITC, and we are just trying to push the sector in this area, in this region, because we are convinced that this is a very, very nice line of work, because we have here sunlight all the whole year around, and we are surrounded by seawater, so the possibilities for the development of algae algal biotechnology sector is very important for us. This is because we are very happy to join you in the promotion and the activities related to the project, just starting from EMBRC, but also to the project, the EBB project. and hopefully we will have new collaborations just going out from this meeting. And remember, we will visit the Spanish Bank of Algae tomorrow, and I think that's all. Thank you very much.
Okay, so thank you very much. That was really very interesting presentation, and so I'll start by thanking you very much for hosting us here. It's already done a very good job, and it's very nice to be here. So, do we have the program? Yeah. On the. So thank you all for coming. Maybe we could start, because I'm not sure everyone knows who everyone is. So maybe we could start just by a few words from each of you about who you are and what you do and stuff like that. Can we do that? So I can start. So I'm Ian Probert. I'm a research engineer at Sorbonne University, which is big Paris university, but we're based in the marine station in Roscoff, which is in Brittany. And I'm in charge of the Marine Biological Resource Centre, which is culture collection, big culture collection, but also all of the aquarium facilities. And that's something we might talk about as well, the aquarium side of things. So.
Hi everybody, my name is Now, does it work? Yeah. Okay. So I was telling that my name is Xavier Lecube. I am the scientific manager of the Biscay Bay Environmental Biospecimen Bank, which is located in the Plenty Marine Station, which is part of the Spanish node, as Juan Luis explained recently. And we are a little bit the freaky ones, because for the moment we don't keep microalgae or macroalgae, but we have other collections more related with tissues from mussels, bivalves, and let's say fishes, marine mammals, and these kind of exotic things in comparison with the rest, right?
Hello everyone, my name is Sergio Sebane. I'm full professor at the University of the Basque Country, and I'm the scientific director of the Basque Microalgae Culture Collection. It's not so big than BEA, but we are also a nice collection.
Hi everyone, I'm Begoña. I'm from the Peninsula, but I did my master thesis I'm doing an internship in the Netherlands. In Al Green is a company for consultancy microalgae, and I did 4 months of internship there, and now I'm here doing the finish of the project in BEA and etc.
Hi, my name is Paula. I am a biologist and I studied in Granada. And I've been working since March in Algae Spanish Bank. I am technical project, and that is all.
Hi everyone, nice to meet you. I'm Tamara. I'm a marine applicant biotechnologist. I'm a researcher of a bank of algae.
Hi, my name is Montserrat Alemán. I'm a researcher at the Canarian Technological Institute, and actually we are working with the production and processing of native microalgae such as spirulina, Chlora vulgaris, Dunalia salina, Tetracermis striata, and a lot of species.
Hi everyone, I'm Martin Gacheneau, and not Martin, it's fine. I'm working at Sorbonne University in Roscoff with Ian Probert on the collection and also on the flow cytometry platform.
Thank you. Hi, everyone. I'm Juliana Carvajal. I'm head of the European Affairs at Polymère Bretagne Atlantique, which is a maritime innovation cluster based in Brittany. So I am a little bit of the outsider of the biobanks. So I'm very looking forward for the discussions today and tomorrow.
Hi, hello to everyone. My name is Mariana. I come from Portugal, Coimbra collection of algae. I'm assistant curator of the collection. It's a collection of microalgae and cyanobacteria.
Good morning. I'm Guilherme, and I'm a taxonomist. I work with cyanobacteria and microalgae. Now, and I'm working at CIMAR in Porto.
Good morning, everyone. Thanks for coming. I'm the curator and the Spanish Bank of Algae and the scientific director. So we are happy to have you here. And I think tomorrow I can show you what we do there. So thank you for coming and hope this workshop will be great and we can discuss and share and develop new project, which is great for us.
Good morning, everyone. So I'm working at EMBRC at the HQ. I'm in charge of the implementation of ABS best practices in our activities.
Good morning, everyone. I am Martina Ferreira from Anfaco Ecopesca in Vigo. We are a technological center that which does, among other things, research in aquaculture. And our role here is more the use as users of marine resources rather than managers. So it's a great chance for me to learn a lot about this topic and yes, make the most of my stay here.
Hello, I am Ivor from Biobank, a marine biobank in Tromsø in the northern part of Norway. I am working with bacteria and
Hi, I'm Kjersti Lie Gabrielsen. I'm a manager or head of Marbank at the Institute of Marine Research in Norway. So Marbank is a national marine biobank, and we hold a lot of different samples from all over the ocean. So it's about macroorganisms and microorganisms. So I'll give you just a short overview tomorrow.
I'm sorry. I am Ignacio. I am the project manager of the Spanish Bank of Algae, and I was the one that have sent all the emails and send you all the all the links and so. And welcome to the University of Las Palmas de Gran Canaria.
Who developed the databases. I know he's online. I don't know if we have other people, do we? Okay, so I don't know how the system works. Are they able to speak? So it's a chat or they can actually speak? Okay. So they can now.
Now we have chat.
They can speak, or it's just chat. Okay. Can we have an idea of who is online? Can we put that on the screen or no? No.
At the moment, no one knows.
Okay, okay. So so there is there is a YouTube going on, but unfortunately the people can't actually speak. If you have chat, you just let us know the questions. So anyone online, just if you have questions, just put them in the chat. Okay, so the program for the next couple of days, which has been sent around. So, first of all, I'd like to say that this is a workshop which is organised in the context of the EBB project, which was an Interreg Atlantic project, which actually ended 1 year ago, more than 1 year ago. And at the time there was a possibility of having an extension, and we applied for the extension and originally didn't get the extension, if I remember correctly. But then just before Christmas we were told that there was a possibility to actually have the extension. That's when a number of the partners, not all of them, but a number of the partners agreed to continue the work, try and finish off some work, and that's where the Canaries people came into the project. It's been a little bit chaotic because of we had Maria in Vigo, who was the project manager, who was already the 3rd or 4th project manager on this project, I think. So she was there, and she was very pushing us to organize ourselves to get done the work that we said that we were going to do. She left in February, March, something like that. And so once she wasn't there pushing us, it got a little bit more difficult to organize things, but then there was the fact that the budget only got validated fairly recently. So this is why we were not unfortunately able to advertise this workshop in advance, and so this is maybe why we didn't have a larger participation, but I think there's enough of us here to be able to do some talk about some interesting stuff and do some interesting work. And what I want to say is that I don't know if any of you were at the EMBRC Community Days. Was it last year in May? And where was it? I can't remember. In Farrell. So for those of you who are there, you'll know this, but we had discussions during these EMBRC Community Days about setting up working groups. And one of the working groups that was proposed was a working group on biological resource centers. So for a start, when we're talking about biological resource centers, for me, we're including everything in there. So culture collections, but also sample collections, also aquarium facilities, etc. So the idea was to set up within the EMBRC a working group on biological resource centers with the objective of being able to exchange on subjects which we have in common, to potentially have staff exchanges, to try and identify if there are aspects of what we do which we should try and harmonize, and things like that. So this is kind of the first meeting of that EMBRC working group. So we're kind of, we're going to be pioneering in this aspect. So one of the objectives of this workshop while we're here is to discuss about this working group and see how in the future we can, what kind of activities we could put together, and see if there is an interest for doing it for a start, but then presuming there is, see how we go forward with this working group idea, and you know, organize activities and meetings and stuff like that. So the agenda for the meeting. So what we wanted to do first of all is, this morning we're going to talk about the work package 4 of EBB, which I was in charge of, which was around amongst other things around databasing, and in particular we're going to talk about the common catalogue of biological resources that was set up within EBB. And the idea with this common catalogue now is, for a start, we've made some improvements to it, so I'm going to present those to you. And the idea of this common catalogue now is that it will be expanded to the whole of EMBRC. Within the next month or two, the idea is that we contact the other EMBRC members who have biological resource centres, who have collections, etc., so that they also include their data. And I think for the moment, most of the people who are here already have some data in there, except for you. So I think it's particularly interesting for you to see this system and see how to contribute to it. So that we'll do a bit of a practical session this morning to show you in detail how to upload data in there and stuff. Then this afternoon, Arno will talk to us about access and benefit sharing. So I don't know exactly what you're planning to do. I guess present some of the work and the tools and the things that have been developed within EBB and within EMBRC. And then we'll also have a discussion about the EU ABS register. So there is a possibility for collections to apply to become to be on a European register as ABS compliant collections. And that's something which I know interests you here, it interests us, and I don't know, we'll see if anyone else is interested, but so we'll have a bit of a discussion about that. And then afterwards, there's a local stakeholders session. I'm not exactly sure how we're going to organize that, but we'll see. It's a bit freestyle. And then tomorrow morning, visit to the BEA, so very looking forward very much to seeing the facilities here. And then in the end of the morning and in the afternoon, talking about kind of common activities, for starting off with more kind of administrative activities, but then also more R&D activities, and seeing where there are common points. And we'll also discuss about there's an EMBRC call for projects coming out, for collaborative projects, so we'll see if there's any good ideas for collaboration within that context. Okay, so let's get going with the. So how do I? Yes. Okay, so this was the work, and I would also like to say that the kind of workshop. So here we're in a kind of a formal setting with high-tech stuff and stuff, but this will only work if everyone contributes and everyone, you know, don't just sit there and listen. I don't want to be speaking for 2 days non-stop, so please, any time you have questions, comments, no question is too stupid. Please contribute, and that will make this a more interesting and a more efficient exercise. So we'll start talking about databases. So I would say that, first of all, I'll say that I was planning to do this presentation along with Fabien, who developed the systems, but apparently he can't talk to us. He can't actually present, so I will do my best to do it myself. He is online, but apparently with this system, he can't actually contribute directly, so I'll see what I can do. So this is the work package 4, which was, as I say, amongst other things, on e-tools for biobank management and user access to EMBRs. So the things we developed within this work package, there was 2 main systems. One of the systems was for biobank management, which we called Track. And this is a web-based database tool which manages data pertaining to EMBRs, is marine biological resources, collected in the natural environment or maintained and cultured in aquarium facilities. And this was something we developed because for collections, most collections have already some kind of database system, and we'll talk about that in a minute because I'd like to know what you have. But so for collections, there already were, in most cases, some kind of tool that existed. But as I said, the biological resource centers, it's not just the collections, it's also other types of activities, and notably one of them, and something we do a lot in Roscoff, is supplying macroorganisms, which either we collect in the local environment and we maintain them for some time in aquarium facilities until we provide them to the users, either on site or remotely. So basically we have a list of species which we can find in the local environment, and the users can order species from this list, and we will go out and collect them and provide them to them. With sometimes they'll be held in the aquarium for a little while, but then we provide them to the users. And the other way to provide macroorganisms is, and particularly for species for which there is a high demand, is to culture them. So we have in our team several people who work on developing the methods for culturing different types of macroorganisms. So in Roscoff we do a lot of ascidians and sea urchins, things like that. This involves long-term R&D to try to be able to do the whole life cycle in the aquarium facilities. And for this activity, for macroorganism supply activity, we basically didn't have any real database for this, and nobody really had a good system for tracking what was going on. At the best, there were Excel files, but often it was just notebooks, etc. Thank you again. Thanks. So this is why we developed this system. And I wasn't really planning to talk about this today. We could go back to it if anyone's interested or if we have time. We'll see about that. If anyone's interested to see it, we could go and have a look at it. So what I'm going to talk about mostly this morning is the database, the system we developed for user access, which is called Trace. And this is a common online catalogue of all of the marine biological resources available from EBB partners. So, Trace, as I said, a common online catalogue, and the important thing to notice first of all for this catalogue is that it includes different types of resources. So it includes resources held in culture collections. It includes cultured macroorganisms. So these are things that which we have in stock. We have lists of what we have in stock. Sample collections, so like the Biobank. But it also includes wild collected resources. So these are things that we can potentially provide. So it's not only things we actually have, it's also things that we can potentially go and find in the environment and provide to users. And the idea of this, of developing this catalogue, this is something which has been going on. We actually, in the first Assemble project, which was 2008 or something, there was an attempt to do this, a catalogue like this already, and it kind of didn't really work. But the idea of this is so that a user who wants to work on a particular species, they have a place where they can go and search for the resource amongst all of the EMBRC partners, and the idea is to have this catalogue which will tell them which partner institute can provide the resource in question. The idea was always to have a system which was developed in EBB, which would be expanded to the whole of EMBRC, and to have a kind of one-stop place where people can go and find what they're looking for. In terms of us, what we need to provide in terms of data is based on a restricted set of data. So this is what we spent a lot of time in the project defining what were the minimum data requirements. And in the end, we got down to, which we'll see in a moment, we got down to a file which contains something like a dozen fields. So you only need to provide data on around a dozen fields, and this is provided by each partner. So with this data, there's possibility to do either manual or automatic data upload. We'll have a bit of a closer look. Maybe that's a good time to see, because the manual upload is very simple. We'll go through the steps of showing you exactly how to do it in a moment. But it can also be, what is much more simple is if you have an automatic upload, and this is if you have already a kind of online system, an online database. We can actually go and make an automatic link to that, and it can go and automatically harvest data. So this is what we put in place for the Roscoff collection, for example. So in that case, anytime I add data into my Roscoff database or modify data, it automatically gets modified in the common catalog. So it makes it much more easy. So maybe it's a good time to see who has what in terms of databases. So maybe you can start.
We have been working very hard on the development of a database for our activities in the Spanish Bank of Algae. And well, Antera is the expert on that, and well, it's based on a FileMaker, database, and yeah, we have been working also how to exchange data from our database to other ones. So it's very interesting to see how it works, right?
So you have an online.
Well, we have in our web page, we have a lot of information that can be helpful for these kind of things. But in our system, we have our own database, which is the FileMaker. But this is not online yet, at least. But in 2 months, I think.
But we have developed this during years just to begin the minimal standard requirements only to show in our web page, but doing it from an Excel file. So now we can do it in 2 months, I hope so, directly just changing in our new database, and it will immediately show up in the web page. We do also that for MIRI, for example. And in this case, it's not automatic, which is very nice to have it like this. We can change it in our database, and you can see it also in EMBRC.
Yeah, so that means you will have there is an online SQL database that will be created if you have presumably if you have the online version, there is an SQL. So that's basically what you need to be able to set up an automatic system. So.
We can see it tomorrow during the visit. We can show, or even today, we can show the structure and how we work on this database, but it's not at the moment. You cannot consult and doing consultancies directly to this database, but we hope to do it in a few months.
Okay.
This is the idea that we can share all this information just automatically through the web page.
Okay. And you have a web developer? Someone?
Yeah. We have a person that is working with this FileMaker program, which is the person that is making the structure and open and close the fields and trying to run it. And now he's also developing the web access of this database.
This is someone internal or a company?
It's almost internal because he's working a lot with us, but it's outside.
Okay.
So
I mean, we'll see but if you want to put automatic an automatic system, we'll need to interact with that person to see what.
Yeah, it would be, I think, accessible. It's very much on that now.
Yeah, so. I understand that. Well, we have to combine what you need to have from the trace database. What are you going to collect from us, and we have to prepare some way.
Yes.
Okay.
Yes, but that it can be done automatically if you have all the fields we need.
Okay.
We'll see that in a minute.
Yeah, because this is we have been working very much on that, and we have a lot of information there. And our objective was this one, that to have as much information as possible available for the people, which is interesting.
Okay, anyone else? Where are you in Norway? Where are you at with your?
Yes, we have an online system called Biobank. It's a commercial product from the Spanish Norway Bio. It's the same as Xavier and those are using. So it's a kind of a commercial product, yes, but it's customized for us and our use and our different kind of samples.
Because for the moment you have put data in Trace, but you did that manually, did you?
Yes, I think so. But I guess it could be done automatically. Yeah. But we have been working a lot with the customization of this database to be able to to share all the different kinds of samples that we got in the collection. And for the time being, we're also working on a catalog with an ordering system. So we're not there yet. So we're really well.
But you have an online system.
Yes, and we will have, I guess, an ordering system as well. So we are quite curious about what will Trace be able to do and so on. So yeah, but it fits in a way. But for us, it has been more into, we're up in the northern part of Europe with all these minimum data requirements and so on to kind of fit in with our oceans and seas up there. So,
well, yeah. Yes, it will.
Yeah.
I mean, it's as you'll see. Well, I think you've seen already. But the data we require is very standard.
Yes.
It's
Yeah.
basically what is the resource? Where did you collect it?
Yeah.
Basically.
But for us,
Where and when?
Trace is and will be an important kind of location where we can show off our sample as well. So we will have to decide upon the information we put in both places and so on. But I guess that it's okay for us for
Yeah,
now.
but the idea is not to put different information.
No, I know.
It's
So
the same.
so it might be that we'll just use Trace. I don't know.
Okay.
We'll see.
Anyone else?
Well, we at CIMAR, we have our catalog in PDF now, but to update, we have to do it manually. And now we hired a company to make this automatic. So we're gonna put our data on a Excel file, and it's gonna be online automatically.
So, I mean, if it's online, and I'm sure Fabien, who is watching, he will probably be jumping because I'm not a massive IT expert, I have to say. But if I understand correctly, if you have an online system, your Excel file gets converted into a XML file, into some kind of format of database, which you would have that online database, which is then linked to your online catalog.
Yes.
So that's coming
very soon.
Okay.
Yeah. Then we have to learn how to link with Trace.
Yeah, I mean, because this discussion is because there are two ways to do it. Either you do it manually, or we can put in place, it's called a web server, where it automatically goes and harvests the data 8 times a day, or you can set how many times a day, and it's the big advantage of that is that then if you change the data in your own database and it automatically goes onto the Trace system. If you don't have that automatic link, you'll change the data in your database, and then you would have to change your input file for Trace and upload it, and so that is a source of error for a start when you're entering data in different places.
But this Trace will be linked with the same database we use for our system will be used for Trace, right?
Yes.
Okay.
Yes. Yeah. The idea is we just need data on I'd say it's around a dozen fields, and you know, if you have the automatic link, you set up the web server, which is going to just go and extract those 12 fields from your online system, and then it does it automatically.
And you can see the Excel we have is like an Excel, but with I think a very is more beautiful interface. But I think we have all your the things that you need in your database. We have almost put in our database. So I think it will be not very difficult to export, because it's an application that is almost free. I think it's a free system to export. So I think in our case it will not be difficult to connect with your database. And I think if you can enter in our collection, you can see it's very easy. It's not very beautiful, but it's easy, and I think it will be very easy to export. So
okay.
Well, as Kirsty said, we share more or less the same kind of program, but I would say that this is important for us for an inner track of the samples, because as I said, we are the freaky ones, so from just a single animal we can have different samples of this animal which need different preservation modes, so you have to keep track of the samples of the same animal. So this program is is perfect for these ones, but at the same time I'm using track as well. I feed the information you were requiring, and I think that it's a very nice window screen, just to because you are in a big web inside the EMBRC, so for anybody getting in, it's very easy to find, and maybe they are interested in invertebrates. It's an easy way to reach to our
we should talk about that, about track later on.
Okay. Anybody else?
No, I think everyone is okay. So when we were developing this system, the idea, I say, was to have a system where you could have manual or automatic upload, and you would have search functions which will allow the users to identify which marine station can provide the species you're looking for, and the geographic origin of the resource they're looking for. An important point to say is, from the beginning, this was not intended as an online shop. So there is no order module within this. It would actually be relatively straightforward to include that, but for the moment at least, this is not the policy of the EMBRC to have a central place where you can go and order, and the order would get dispatched to the different nodes, or potentially in the future that could happen. I don't know whether it will or not, or whether that would be useful to do that, or I say so this database really is just for going and finding, and then you have a link to the partner which provides it, which makes it a little bit less user friendly, because if you have a user who wanted to order same species from two different partners, then they would have to go and do their two orders separately via the systems that exist. So I say, but that was the policy for the moment, and we'll see if that in the future, whether that changes. Okay, so minimum data requirements. This is actually not completely up to date because we've got rid of some of them. But as you see, there's around a dozen fields that we need. The most important one is obviously to have a unique identifier, so a code for each resource, and I think everyone knows what they are and has those things. We did have some debate about whether originally we did have some debate about whether we were going to make a unique EMBRC identifier, because there is a very minor chance that somebody would use the same, like we use RCC, which is Roscoff Culture Collection, so RCC 1, 2, 3. There is a minor chance that what's another town beginning with R? Can't think of a town beginning with R, but there is a minor chance that someone else would come up with a RCC as well. But we decided that that was not likely. So you use yours is BEA, I guess. Yeah, so BEA1, etc. So you'd use that as your unique identifier. The worms ID. So I don't know if you have this already. You know what everyone knows what worms is? Yes? No? Yes? Maybe. So worms is the World Register of Marine Species. So it's basically a big long list of species names with each species name has a code. So what we do is we use this to automatically import all of the taxonomy. So this means you don't have to add in division, class, etc., because that's a way you can get errors. It means we're using the taxonomy from worms, which in some cases I don't agree with, but that makes it much easier to do it that way. If you don't, or do you already have worms ID in your database? Because we'll show you there is a very easy way to include it. Originally, I thought that everyone would have to go one by one, find the species name and find the number. And there's actually a very useful tool on the worms site where you can do this in a batch. So you can just put your whole list of names and it will give you the numbers. So it's actually a very easy way to do it. We include species name, so that's genus species. That's actually not really necessary, but it's just to check that there aren't any major errors between the worms code and the species. But technically, we don't actually really need that. Actually, the only field which is obligatory is the code. All of the others, if you don't have the data, you don't have to add it. Obviously, if you don't have a species name, it kind of makes it less interesting, because no one's going to search for something which you don't know what it is. So, but it's none of the rest is obligatory. So other codes, that's if you have a lot of, this is more for culture collection stuff. If you have a strain which has an equivalent code in another collection, whether it's a type strain or not, and then the rest of the information we need is about basically the sampling. So the date, the country where it comes from, the coordinates, so geographical coordinates in decimal, and then the ocean, which comes from a fixed list, the region, which is not a fixed list, and the site. So that's specifically where it comes from. A habitat type from a fixed list, and that's I can't remember exactly what that is, but it's like benthic, pelagic, or something like that. And then the last two, ABS status and ABS status remarks, we don't actually need those anymore, and I'll explain why in a minute. But that we've improved the system, hopefully. Arno will tell us if you've not seen this yet, actually. So we just, in the last week, Fabien has made some improvements to the system. So basically now we don't need the ABS status or the ABS status remarks, because that is generated automatically from the country and the date. So we have a table which has all of the countries and their ABS status, and so it just goes from the country and the date that you put in, and and automatically generates the ABS status. So that makes it easier as well. So as you see, there's what, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 fields. It's very, very simple. We really don't need much stuff to be able to contribute to this system. ABS status, maybe we'll come back to that. Well, I mean, this is where we were at with the ABS status. I say that we've made some changes, which I haven't actually completely looked at in detail yet. So we'll discover them together, Arno, and you will tell us whether this is okay for you. But basically, the ABS status is so which you don't need to provide, which will be generated automatically, but it's a response to the question: are users allowed to use the biological resource without applying for formal authorization? So you understand that question? So it's basically what we're providing to the users is an indication with all of the disclaimers, which I'll talk about afterwards, our appreciation of whether when somebody orders this resource, they have to do some application to the country to be able to use it. So basically, the reply: are users allowed to use the resource without applying for formal authorization? If the answer is yes, we've got this traffic light system, so green, orange, red. So yes, that means that our analysis shows that the users can order the resource, use it without having to apply for anything. If the answer is and very often the answer is no, they can't, and even if we're not sure, we say no, you can't just use it freely. You have to contact the ABS NFP is national focal point, so it's the person in each country who is designated to answer questions about this and stuff. So, and then I can't remember if we still have no red. I can't remember whether we still have that. I think it's most of them are either green or orange, basically, because the answer no, contact the ABS. That means that that means that they may be able to use it, but they have to make some application into the country, who may say yes, who may say no, but. And then we had the ABS status remarks, and I can't remember, Arno, whether we were still keeping these. I think we are.
Thank you. So the remark was to explain why we chose the color.
Yeah.
Is there an exception?
Basically, so the ABS status remarks was to give further information on why we think that, for example, if the answer is yes, which means they don't have to apply for anything, there are several different reasons why it could be. It could be because the country of origin is not party to the Nagoya Protocol and they have no national legislation. It could be that the country is a party to the Nagoya Protocol, but they've decided not to put in place any national legislation, etc., etc. So it was to give more information on how we came to the conclusion that it was either yes or no, basically.
Exactly. And what is important today is to know if these ABS status are clear for all of you.
Yes.
Because it will be on the website and
yes.
for users of biological resources, and it's we need to be clear.
Yeah.
So we need to know if we show these definitions or if we just say
yes, no.
Yes, no. What we need to.
So that's definitely one thing we'd like you to do this morning is to go onto the, if you have your computer, go onto the site and look at this and see whether you think the way we've done the ABS is confusing, or is there too much information, not enough, or, you know, because we don't really need to add the ABS status remarks. I think it's useful. It gives a, but are we confusing people too much if we add that, basically?
This information will be on the passport, so we will provide it when we provide the bioresources. But do we really need to put it on the website? That's a question. And we have time to talk about it this morning, but also this afternoon, because I also plan to talk about it.
Okay. Yes.
Thank you. Thank you for the explanations. It's just that this part is very linked to the users, so our members in a cluster. And I just wanted to know, because indeed for research development, we know that if you have already all the ABS authorizations, so you can just give the resource to the enterprise, and they will work with it, because it's research and development. But once they develop a commercial product with it, they need to pass to the other, the other process about the commercialization of the. So I wanted to know if all the biobanks here, because I know that you were working in all the Sorbonne University ABS authorizations. So I just wanted to know the current status of that. So all the biobank has already the authorizations? Or
No. In fact, the way ABS works, we cannot, a biobank cannot ask for, and you correct me if I'm wrong, but a biobank cannot ask for ABS authorization for a user. It is the user who has to ask. And for each use, each different user has to ask. We actually looked into whether, as a resource provider, we could make an application saying, okay, we're sampling this resource. Can we have authorization to provide it for any use? And it doesn't work that way, in fact.
Okay,
unfortunately.
Yeah, because in that case, I think that for the industries, trades will be interesting maybe to get the resource, but if they need to themselves to apply for the authorizations, this needs to be very clear once they get the.
Yeah, I mean, what will be interesting for them, I think, would be to choose species which are green, yes, where they don't have to apply. I mean, it's maybe a bit more complicated than that for commercial use, because countries which have said there's nothing, is the same for commercial use? That was a question.
Sorry, just to say that a quick reaction on that, because in that case, even if it is not commercialized, but it's only for research and development, I know that for enterprises would be nice just to skip that. But if they need to do it, that's.
Yeah, but I mean, technically, if it's something which is green, they could skip it. Yeah, that means they can look through, see who can. You can put that as a search criteria. So I want this species with this ABS status. So they could say, I want this seaweed with the status yes, so they can use it freely, and so that way they could see who could provide them with these resources.
So I can confirm that the obligation to obtain the authorization is a user's responsibility, not the biobank's responsibility. For commercial purpose, you might need authorizations when it will be in green, in orange, but you can choose bioresources that we identify as in countries that do not have ABS restrictions. For fundamental research, we have obtained a first example of, is there a possibility to transfer the responsibility on biobanks? There is a possibility to obtain bioresources from a register collection. We talked about it this afternoon. And also we have obtained, I would say, in the Genomic Observatory, EMBRC in EMBRC, we obtained the authorization from France to do the due diligence for users, for internal use. So EMBRC can do this part for users. But
yeah, I think in fundamental research that will be more and more the case. You know, for a project you'll be able to get you know, an authorization saying for the users. So but I don't think that will ever happen for commercial users.
Yeah, this is what we want in EMBRC. We want to show that we have the process to monitor everything, and it will help us to negotiate these kind of things. But it's possible for fundamental research. For commercial research, it will be more complicated because there is some issues around appearance.
I think we had a coffee break scheduled 10:30. Is that right? So that would be probably. I just want to show one more slide actually. So on the site, and we'll after the break we'll go and have a look at the site. But so we have a page which is an ABS disclaimer, which explains the system. So I'd really like you to go and read that and see if you make sense of that. And then this is the new thing that we've just put in. And we need to talk with Arnaud to see whether this is the way you imagined it. But basically now we have this, so that it's actually as a page. This is from the table. Arnaud provided us with a table saying this country has this status for this reason, basically. And this is the way we've put it in place. So you have a page which has all of the countries going down, which explains the status. You can search for the country, and as editors, Arnaud and myself, but mainly you, can actually then, if there is a change in the status of a country, you can go here and edit the page, the country in question. And when that gets edited, then that would automatically update all of the resources in the collection. So this is a much easier way of doing it, I think.
How far can we go on there are for countries? The status as for countries. We can say, for example, in France, we know you might need a declaration or authorization, so it will be in orange, but you need to contact the ABS National Focal Points in this country. But we know that sometimes there is exceptions, like microorganism might be an exception in France.
Yeah, I mean,
so
we could, I think,
how far can we go?
I think in technical terms, and Fabien, who is online, he will probably curse me if I say this, but in technical terms, that should be very easy. We could say, you know, if it's from France and it's a microorganism, it's green. If it's from France and it's a macroorganism, it's orange, because you can pull out from the taxonomy. You can make and we already have done that actually. We've based from the taxonomy, you can pull out whether it's a microorganism or macroorganism. It's not 100% efficient. But so yeah, I think you could go into more detail if you wanted to.
Okay, because we need to talk about this later, because for now we just want more basic system to say we are sure this is green, and if it's not green, then it's orange. But later we might have a look to specificities to make the more resources in green.
Yep.
Okay, so we talk.
I'm glad to hear you say that, actually, because
yeah, that's the plan.
Yeah, good. Okay, let's have a coffee, and then afterwards we can go and look at the way to upload data and stuff like that. Okay, so we'll get going. So Fabien who's the web developer. He's actually on Teams now, so he can help me to show you how to do the upload. So hello, Fabien. Can you hear us?
Hello, everyone.
Yep. So I'll start off by just showing you the system. So you can have a look yourself. The address is trace how do you call that? Hyphen EMBRC .eu. So what we have in the system, so it's very simple system. On the first page you have a little blah blah about what the system is, which explains that it's for finding biological resources. And on the first page you have a quick search. So this is the first thing you see. So give me a name of a species. Something more exciting. No one has a species.
Mytilus.
What?
Mytilus edulis. The muscle. Why not? Not only a microalgae.
So you type your name in. It will give you the. It will show you the matches, and then you press go, and what it will come up with is a table which will show you all of the resources that are available in the database for Mytilus. And in this first table, so you have the code of the sample. You have the basic taxonomy. You have the origin, the ocean where it comes from, and the country. You have the resource type. And as I said before, it's important to notice that in the database we have different types of resources. So it's basically wild or cultured. So wild is collected. So it's a sample collected in the wild, or it could be cultured. The first important thing to know is you can't put wild and cultured resources into a same file. They have to be in separate files. I'll explain that afterwards. You have the ABS status, and then you have the link to who provides that. So if you click here onto the code, it opens a page about that resource in particular, and this is what is called the passport of the resource. So it shows you who provides it. It shows you the ABS status with the explanation of why. It gives you a link, and I think maybe Arno will talk about that later. It gives you a link to the ABS clearinghouse and to the EMBRC step-by-step guide on ABS. So you'll talk about those later, I guess. Yeah. So I mean, it's a link to resources about ABS. Then you have this. It tells you different information about the resource. So this is a wild collected. It's fixed, so you have the samples are either fixed or live. So that's something that information that you'll put in at some point. We'll show you that. On-site or remote. So that's some resources can only be provided on-site. In the marine stations, and remote is if they can be sent as well. So the two categories there are either on-site only or on-site and remote. So it's not a type strain and it's an invertebrate. So we have the different categories of organisms. I say that's taken from automatically from the taxonomy we've kind of designated that such and such a class belongs to invertebrates or algae or stuff. So you can later on, you can search by all invertebrates or something like that, but that's done automatically. So it gives you the identity, so the taxonomy. And as I said, this is all taken from the worms number. It's automatically taken out of the worms database, so it gives you all the taxonomy. Actually, I don't know what happens if you click on those, but we'll see in a minute. And it gives you a map, it gives you information on the origin of the samples, so when was it sampled and where, so the different information on which ocean, which location, which region and which site. And it gives you, if you've put in the coordinates, it gives you a map with the location and the sample habitat type. And you can upload pictures, and we'll explain that afterwards. So that's the simple search on the home page, the quick search on the home page. You also have a couple of other different ways of searching. So there's advanced search. So this gives you the possibility to do a search with multiple criteria. So let's look for, I don't know, something for what do we have? A bacteria, Alteromonadalis is a bacteria, I think. So if you just do one, it will give you the whole list of the organisms which match that criteria. But then you can also like refine your search, saying I want Alteromodius only from the Arctic Ocean. And then you're down to 5, and then you could also do, well, you can do as many criteria as you want. One comment actually for Fabien, which we had later on, is we don't actually have the ABS status here. And it would actually be quite good to add that if it's possible. So we can search by ABS status as well. You have a comment, Fabien?
Yes, about the ABS status, this is not built yet for this search because I am waiting for your decisions about the remarks and if we just make something very simple with yes or no. And as soon as this decision and this discussion is okay with you, I will just build this for the advanced search.
I think probably on the advanced search we'll just have the yes, no, probably, but
yeah,
we'll see.
Okay. Probably the icon, I guess.
You can mess around with your search and take out criteria and stuff like that. And so that gives you the same table where you can go and click on the resource. I didn't mention that in this table, so it gives you the resource provider. And if I click on this one here, it will take me, if you have the website which has the a page for each code, it will take you directly to the page, in this case of the Roscoff collection. So it's the page of this strain. So you click on that thing and it takes you directly to that page.
And if I just can add something?
Yes, yeah. Of course.
About the details for each resources, I will need something special. This is a specific address, because, for example, on the Roscoff Culture Collection, each strain has a specific pattern that is repeated for each strain, something like RCC strain details / and the RCC number. So if you have something like this, I think that the CCAP has something similar. If you have this specific recurrent pattern, I can just use it to automatically link the resource from Trace to your website. But I need this.
So that is the address up there, the RCC strain details 2408. So if you have that kind of system, you can link directly to the page. Otherwise, the link will just go to your home page.
Yes, exactly.
We can do a link just to your home page, but if you have that system, you can go to the actual page of the resource itself. Okay, and the other way to search. Yes.
Where this data uploaded manually? All this data that you are showing us?
Which data?
All this information that you get from there, were all uploaded manually from every laboratory? Or?
Well, no. For the moment, the only one which is automatic is the Roscoff one. So basically, I don't do anything. We enter our data into our database in Roscoff, and it automatically gets updated onto the thing. That's what we were talking about this morning. For the moment, all of the others, or did we do that with the CCAP? I can't remember. We did automate.
No, they made a manual one.
Wow. There is a lot of information from the CCAP, so they make a lot of work.
Yeah, and it's going to get kicked out because they are no longer members of EMBRC. But I mean, as you've seen that the table, the information we ask is only a dozen fields, and it is actually should be fairly easy to extract that from your database. So it's actually not a huge amount of work.
If I may say something about this, Ian?
Yes.
Yeah, about the automatic upload, because I heard you this morning about this, and it's okay.
I was saying stupid things. Yes.
About automatic upload, for example, for the RCC, on the website, there is an automatic extraction of the minimum data requirements for trace, and it's done automatically once a week and not 8 times a day. Okay, so just once a week, and a CSV file, a specific format that we will see later. A CSV file is produced every week, and the Trace website is fetching this data once a week too, during night, and using this CSV file to import the new resources onto the website and to update if needed. So the only thing I need if you want an automatic upload, I heard that some of you need this, is that your IT or the people in charge of the website just create this CSV file accessible online, so I can just interrogate this file, and this is called API. I suppose you are familiar with this system. And so if your website is producing this CSV file, I can just interrogate it and question it to fetch the data and use it automatically.
Okay, so I mean, for that, basically, you would need to put myself and Fabien in touch with your IT person, and we can work that out.
Yeah. And just to finish, it could be CSV or maybe XML or JSON. That's some typical format.
I'm not sure that any of us here understand that.
Okay, so I am here to help, no problem.
Okay, so, and there's the third way to search, which is the map search, which takes a little bit of time to load at the beginning, but that's because there's a lot of things in there. So the map search is basically the same search, but what it gives you is two maps. The first one is where so if you ask for, so let's ask for a, I'll try a different one, Focus. So it gives you, the first map shows you the geographical origin of where the resources that are available actually come from. So we have some focus which come from Roscoff and some from, who's that, Porto.
You can click on it to have details. Click on the marker.
Yeah, so you click on the thing and it gives you the code and a little bit of information. Then you can click onto there, which will take you to the page. And if you have more than one which comes from one location, and you have these three little dots and you can click on each one. And it gives you the second map, and actually this was not a good example because it's exactly the same, but it gives you shows you the location of the stations which can provide the resource. And we have the two maps because it's actually more for cultures. I'll try a different one. Let's try a good old.
Just reset your search.
Because I mean, often the resources might have been isolated in the middle of the Atlantic, and they are available from Roscoff. So you have the two maps which show the different things. Why is that spinning? Why have I got the spinning wheel?
Now it should work. Maybe you can just refresh your page.
So, any questions on the search functions and things like that? And also, I mean, the point of showing you this is also if you have any ideas to improve it. We're always open to ideas for anything that might be missing or is too much. Let's have a look for one which I know that we have lots of things from all over the place. So there you see on the map for the geographical origin of the strains, we have ones which come from a bit all over the place, and then on the map you have the different stations which can provide them and how many are available from each station and stuff like that. So it's a way to find out if you want to search by the origin of the resource or where it comes from. Okay, so afterwards the other pages, there's a page on statistics. So that, I mean, we put in a few different statistics. It's on the, I think this is, it's quite nice. Once everyone's data will be in, it will be quite nice to see, EMBRC at the moment. Well, this is just EBB for the moment, but you can see the biggest in terms of taxonomy. What we can provide is chlorophyta, etc., etc. And then we've got one on the resource provider, the number of samples or strains provided by each provider, the country of origin of the resource. So at the moment, there's a quarter come from France, but this is obviously biased because of the Roscoff data for the moment. And then we also have ABS status. So you can see that for the moment, 80% of what we have in there can be used without asking for authorization. So
it will be updated.
Sorry?
It will be updated with the new ABS status country.
So that's some stats. We could put other stats in, I guess, if there's any other stats that anyone thinks would be worthwhile. Then we have the ABS disclaimer, which I showed you earlier. So at some stage, I'd really like you to read this and see if it makes sense to you. I mean, particularly Arnaud, but the rest of you as well. And the point about this disclaimer is is really to say that this is, we're doing this to try and help users, but we're not responsible if anything is incorrect. So it's important to have that disclaimer there. And then this new page on the ABS compliance. So we have all of the countries here with the situation pre-Nagoya and post-Nagoya. Yes.
Microphone, please. If not at home, don't hear you, please. Thank you.
Hello. Wait. Is it possible to include in this database if the strain is a type strain or a reference strain?
Yeah, it is in there.
It is in there?
Yes. Let's have a look. I think of one which is a type strain. Even if it's not a type strain, in fact, the information is there. That was one of the criteria for one of the data fields is type strain. So when you go to the page, it doesn't show on the first page, but it on the passport for each. Actually, it doesn't appear there. Yes,
yeah.
It does. There it's
yes,
type
it's
strain.
Yeah, but in this advanced search, if we want to search for type strains or reference strains.
Yeah, we could add that, I guess.
Yeah, it's noted. Yeah, no problem. It's noted. I will add it.
We can add that. No problem. I don't really know why we didn't put that in there in the first place. But.
Yeah, me neither.
Okay, so now we'll get to, so that's basically the system. And now we get to how you upload data. So I don't know, Fabien, do you want to take over or shall I try or?
As you wish. Maybe this is better that you try first, and if you have problems, I can just intervene.
Okay. So for all of the data providers, there's a system to log in. So you'll have a login. So for anyone like for you, we would need to know who is the name of the person who is going to do this. And I guess it can be more than one person, can it, from one institute, or is it just one person?
Yes, no, we can have as many people as you like. No problem.
So we'll need to know. I think for the moment we put Ignacio, but
not yet, but it will.
He's not here. No, you don't need him. It's okay.
I can just give you something. Do you want me to do this? Because I think I have everything in place here. So if I can try to share my screen, I think it will be easy.
Can we go to the Zoom? How do we see the Zoom Teams? Okay. And can you share, or do I need to do something?
I think I can share something, but I hope that the connection is all right for you. Do you see something nice, or is it a little bit blurry?
We can see you for the moment.
Okay. Hello. Okay. Let me see. I have to give access to Teams. Okay. Right. So, do you see something? Oh, no. Yes. Do you see my screen? Yes. Okay, great. Do you see the Trace website? Yeah, okay, good. Okay, so first thing to do, of course, is to log in, just as Ian said. And just for this demonstration, I will show you how to import data. I have just made a fake account called the FCC Manager. FCC for Fake Culture Collection. Okay. So just stop me if you don't get something or if it's not clear enough. Okay, I will just explain it again. So when you're logged in, you can have access to this very simple page, and you have here a very short.
For the moment, we're still on the login page.
Oh, okay. So we have some lag. Okay. So I think you see something. Oh. Okay. So do you see the FCC Manager account? Yes. Okay. On the right side, you have a Trace Manager Tools menu, and you have here one link to the FCC Cultured CSV import. So this is something that Scroll built for you. You have nothing to do here. And as the FCC Manager, I can only see this one and not the other one, so I cannot import data to other collections. So this is safe. Okay.
If you have, which is the case, for example, in Roscoff, we have cultures, but we also have wild collected things. So there I would have two files. One which is called RCC cultured, one which is, I've actually got 3 files, because for the macroorganisms we have cultured or wild. So I have one which is macroorganism cultured, one which is macroorganism wild.
Exactly. And so for this purpose, I have to create 2 different import procedures, and you have to provide 2 different files. But it's okay, and I can explain everything for each one of you. Do you see now the import page? Yeah?
Yes.
Okay. So on this page, you will see some previous data if you have already imported things. And at the beginning, I am the one who built this, and I always imported a short data set, maybe 5 or 10 resources, just to start the import procedure. And after that, you can just import as many strings or resources you need. And on the right side, sorry, you have a few indication and information. And the most important is the template file here. Okay, so you have a menu with Excel template file, and another one with a CSV file example, and the Worms taxon tool that Ian spoke before. And maybe you will just show this later, I suppose.
You want to show it now?
Actually, I have never used it, so if you are more familiar with this.
Yeah, I'll show you the Worms tool later on.
Okay, good. Just to finish on this page, if I just scroll down a little bit, I have prepared yesterday a video tutorial just to explain everything I am going to explain now. So don't worry, you don't have to know things. Everything is in place in this video, and I hope it's okay to follow this for you. It's online on YouTube, and it's always available on your import page. And just scroll a little bit, and you have also these import guidelines with 6 main steps to follow to import correctly your data. So you can just follow these steps, and it should be fine. Okay, I will just scroll up, and I will just show you the Excel template file, so you just see what Ian presented before, the 13 columns and fields. So you just have to click on this link, and you will immediately download the template, and you can open it on Excel. I will just zoom a bit. Okay. Can you see the Excel file? Yes, okay. So it's okay for the lag, it's maybe better now?
Bigger would be good.
I can't do anything, actually. Okay, so oh, bigger. Okay, sorry. This is correct. Yes. Okay. So to be honest, I think that the worms ID could be required too, because sometimes I know that you have strains and resource that has not been determined and defined, so it could be difficult, but it's really better to expose, as you said, a catalog with known species. I suppose
And obviously if you don't know if you don't know the exact species, you can put just the genus, because in the worms there is a number just for the genus, or even you can put just eukaryota, for example, if you wanted. There is a number for that, so you don't have to have the characterization right down to the species level.
Exactly. So I guess that we can say that it's highly recommended to have the worms ID, I say. Okay, so for the code, okay, no problem, it's required because this is something we call the primary key. Maybe you are familiar with that kind of system in the database. And so we need this code absolutely to distinguish resources between them. And after that, you see that you can just enter the data, just the simple one, species, code, type strain. So you have, on the second line, you have some guidelines and information to provide the right format for this data, because if you put yes or no for type strain, for example, it won't be imported correctly. I need something with 0 and 1. This is informatics. So we have to work on binary system. And for the sample date, it's important to have that kind of format with the day, the months, and the year separated by hyphens. Okay. And these two columns, dates and country, are really important because these two columns will be used to define the ABS status and remarks automatically. So you don't have to wonder if it's yes or no. We will decide for you, but we have to know when and where it has been sampled.
Yeah, but if you don't have that information, what happens? So if you don't have the country, it gives an ABS status, which is...
Yes, we have a special, we discussed this. This is no and G, if I'm correct, so no red, and because we have no information on country. And so if you don't have anything on sample country, there is a default procedure, default system that will just give this ABS status specific.
And the same with the date?
And for the date, I think we have to discuss this with Arnaud too, because I suppose that if we don't have the date, sometimes it's very old resource, old strain sample, maybe 1958 or something like this. And so this is something called, that's pre-Nagoya protocol. So I guess this is the same thing, even if we don't have the date. But I don't know.
I can see Arnaud, yeah, he can't hear you, but we'll talk about that later.
Yeah, okay.
But anyway, if you don't have the country or the date, there still will be a status, but it will more likely be a red.
So you should have a red if you have no country. No date, I think I put automatically the pre-NP status. And if you have neither no date and no country, I think you have just a message to say that you have not enough information.
Yeah.
Okay. So the coordinates, and of course you have to put this in decimals, okay, not degrees, north, and etc. And
so it's minus for south and for
yes.
East.
Yes, yes. Negative values allowed for south and negative for west for the longitude. Okay, it's here. It's written here. Okay.
Yep.
Yep. So when you work on your data on the sample on the Excel file, just the end is just something typical, nothing to say here. But when you work on the Excel file, the first thing to do is to check if everything is in place and the right format. And you see, for example, this is a mistake on purpose. I have put this coordinates with a comma and not a point. And if you let this like this, you won't have the map on the resource. So you have to correct this and just point for the decimal, and it's okay. So that's the kind of thing that is currently generate errors. So just take good care of this data. Second thing we can check, and it's when you just go and copy and paste things. A lot of time you can have some unexpected spaces on these different cells. For example, here you can see something, but if you just select everything, you can see that at the end we have a space, and it's not really useful here, and it will not be filtered as the other data possible, Western North Pacific, without this space. I don't know if I'm clear here, but you don't have to get this space because it won't be filtered at the same place. Okay, so you have to delete this space, and it's okay. You can just check this here too. Okay, and sometimes you can have some extra lines at the end, and you have to check this too because it will import empty resources, and it's not something clear and clean for the website. So if you already have a database with many different things, you just have to fill these columns, and you're done, and you're okay to import data in the website. So when you're done here, you can just, of course, save your file with the right name and so anything you want. And the second step is to save a second time, but in another format, the famous CSV UTF-8 format. So you have to save as, I'm sorry, this is in French here. And you have to check and to choose that specific format CSV UTF-8. This is really, really important because the website is built with Drupal, and Drupal is always reading very well the CSV and not very well the XLS or something else. So it needs that kind of specific text format, and especially the UTF-8. UTF-8, it means universal text file, and it will take care of all the specific characters that we have in many different languages. In France, we have a lot of accents. I think we have some accents in Spanish too. And everything will be take care. And usually with this format, it's okay. Everything is well imported. Okay, so you have
just a question. So all of the Excel versions have the possibility to save in CSV UTF?
I guess that maybe the older one can save only in CSV and not UTF-8. But I will show you the next software, Visual Studio Code, and I will show you how to encode in UTF-8. Okay. And this is a free software from Microsoft. This is something we can notice. Okay, so for the demonstration, I have prepared two different files. Let me see. Okay, yeah. Okay, so I'm going to work with this file. Okay. So it's called FCC data May 23. And just I am the curator of the FCC. And I want to import 20 new resources within the website. Okay. So I have my code, FCC1 to FCC20. I have all the ones ID. And I have just put a few different things. Actually, this is some resources from RCC. and maybe Roscoff Aquarium Services. Me. Okay. Something really important to remember that on the second line, we have some information on format and how to enter your data in the Excel file. You have to delete this line, of course, because we don't want to import anything about this line. And it will indeed make some mistakes and errors. So you have to delete this. Okay. Just to remember, let me just show you this. Okay, this one. You see this line with all the different things, you have to delete it. Okay? Oh, okay, sorry. So this is the template I have downloaded from the website. And the second line, the number 2, has to be deleted each time. Okay. Yeah, okay. So remember, this is the template and the second line has to be deleted, okay? It's just a guide for you to enter the data, and once it's done, you can delete it. Okay, so I'm going back to this example. And from this Excel file, I have prepared a CSV file. And now I'm going to show you this with a specific software, this one. Let me just do this, okay. Okay. So can you see the new window? Yes, something darker. Okay.
So that software is called what?
I'm going to show you how to download it. It's completely free, so you can just add it today if you like. And this is called Visual Studio Code. So this. Okay. This is, can you see the web page now? Yes. Okay. So if you go to code.visualstudio.com, you will go to this page, the same page, and you can download it for Mac or Windows. No problem with the two different computers. And this is something like we call IDE for Integrated Development Editor. So we can make a lot of different difficult and complex code, but you can also simply open CSV with this software, and it can help you a lot to see if the CSV is okay. So we just go back to the VS Code window. And the nice thing with this software is that you can just add some extensions, some plugins. And one of them is Rainbow CSV. This one. Okay. So you have something on the left side with some Tetris cubes here. And this is called extensions. You can just search for Rainbow and you will just find this. This is really famous. It has been downloaded more than almost 3 million times. Okay. And this extension can make you some highlights on your CSV. As you can see, we have now colors on the CSV here. And if you just go maybe on species here, you see that this is the third column. And if you go on the same color, it will just remind you that this is the species. So if you see a country in this column, you have an error, okay? You have something to correct. First thing. Okay. The second thing, I will just maybe do this, okay. And the second thing is that this extension can check your CSV, check the integrity of the CSV. And what is this? Sometimes one record can be displayed on the CSV on multiple lines. And if you have that kind of problem, it can happen when you have some back return on text. It won't be imported well in the website, so you have to check this. And the extension make it for you. You have just this little thing here, CSV int. If it's green, it's okay, you're done. Everything is fine. But it's not. But it will be checked every time you open a CSV. And if it's red, you have to check the different records where you can have some multiple lines, and the software will show you where it is. So it's really helpful. Okay. Yep.
Just one comment. You don't have to do this if this seems too complicated. First of all, we can do it for you. Or secondly, if you've done your Excel file well and you've saved it as a CSV, there should be no problem. So you can go directly to the next step. It's just then when you upload it, you have a problem, then you would have to come back and check. But you don't have to do this.
Absolutely. Is it a bit frightening? Okay. So we will go back now to this. I will go back to the Trace website, and we will use this CSV file to import the 20 resources from the FCC. Okay, so can you see the Trace website? It's okay? Yeah, okay. So back to this page, you will have, you know that I am logged in as the FCC manager. So everything you can see, you can just use it, of course. And you have some different tabs here. Okay, we are on view at the moment, and you can edit this procedure, this import. So we'll just click on it, and you open a small form. Okay.
Hang on, we still have a bit of a lag.
Yeah, I'm waiting. No. Okay. Okay. On this form, yeah. Tell me. It's okay. On this form, you just have to work with one thing. This is the file here. You can see that we have something already imported, but this is only test I made yesterday to be sure everything is fine. So I can remove this file. I don't need it. Okay. And after that,
every time you update your data, you remove the file that was there previously.
Yes, because if you imported new things, it won't be on the previous file, obviously.
Yeah.
Okay. So you remove the previous file and you choose your new file, just the one we made before, on your computer. Okay. And from there, you don't have to modify anything else, the delimiter, the headers, and everything like this. You just have to change the file and finally click on Save and Import. Okay, so I will just click on it. I hope that you will see the little progression, the progress bar. I hope so. Okay, so I'm going to import my new resources.
Yeah, okay, we can see it.
Yeah, okay.
Yep.
Okay, it's done here. Just to say that it's not too long. Can you see a green message?
Yes.
Okay, so maybe you can just zoom a little bit here. Okay. And as you can see now, we have created 20 resource items only by using this CSV file and just import the file at the right place. Now I'm going to check if everything is okay. So I'm going to the advanced search. Okay, just tell me when the search is okay. Yeah. Okay. And just to look quickly if everything is good, I'm looking for the resource provider, and I can see here scroll failed culture collection with 20. So I think we're good. We have the 20 new resources. I'm going to look at them. Okay. So you can see that I have my logo here. So I didn't put this on the CSV file. This is something automatic because I am the FCC manager and I'm part of this institution. Okay. And if you just open any genetic resource, everything is in place for the ABS status because, as you can see, this one was from France and it was sampled in 2002. So this is pre-Nagoya protocol. So we have yes, green, and C. So I suppose it's okay. I hope Arno is okay with that. And automatically we have the ABS clearinghouse. And if you click on it, it should open the page directly for France. Okay. Can you see the ABS clearinghouse? Yeah, okay. Go back here. As you can see, I have entered only the worms ID, and I have all the taxonomy here. So no problem with that. And the map is okay. It's done with this kind of coordinates. Yes, 48, -3, I think it's okay. And what about the category? It's automatic too, because I have built a little code that will just look inside the taxonomy and put the resource in the right one. Now I will show you something. I will show you when we edit a resource online, because you can do that too. Yeah, sure.
Is there any questions about the upload procedure? Does that seem...
There will be a second one, just to see what happens when we just change things. Okay. Yeah. So if you see something that you want to transform and to modify directly on the website, it's possible to. You just have to click on Edit, just below the title. Okay. And you will just open this form. Is it visible? Yeah. 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 Just the time to drink some water. Is that okay? No? Oh. It's good. Okay. No? Okay. Okay. So you see FCC 10 Asterias Rubens. Okay. If I just scroll a little bit, you can see the taxonomy worms ID, 123, 776. Okay. And maybe just take another one, this one. If you just to show you how it is, how it's automatic. If I just change the worms ID here, for this one, 235923, and just go back to the previous field. Just look at the taxon field just below, and you will see that they will change automatically. Okay. And everything has changed to a Calcidiscus. Of course, this is not the right species. It was just an example. No? Okay. And you can see that this is Calcidiscus. Okay. And if I just go back to the previous worms ID, it will change and go back to the previous species. Okay. So that's why the worms ID is really useful for us. Is that okay? Yeah. Okay. Second thing we can change, and you can change everything actually. You can just download image too here. Nian told you this before. You can just select files from your own computer and just put anything you like with that kind of types, PNG, GIF, JPEG, etc. And I have put this limit at 64 megabytes, but if you need more, we can just increase this limit, of course. Okay. And here you can see that we have a second tab called Origin. If you click on it, you will have the second part of the form. Okay. And here, remember that we have yes and C for the EIBA status. I will just change the country, for example, and put Italy. And I will save the resource because it was a mistake and I just want to change this. Okay. And when you save the resource, it will automatically change the ABS status. And now if you can see the resource page, we have now yes and A because Italy is not part of the NP and no ABS authorization is required. Okay. And that's the same for the sample date. I'm not quite sure this is useful. I show you this because this is the same thing. You can just change. And for the moment, I'm not sure it's okay, but it was the start for us, for Ian and me. The switch between pre-Nagoya and post-Nagoya is 2014. Is still correct? Yeah. Okay, so if you just put the sample date before this year, you will have a pre-Nagoya protocol ABS status and remarks, and if it's greater than 2014, it's post-NP. Okay, and of course, everything can be changed later.
I have a question actually about, so
yeah.
you can upload images. What happens if you then later on update your file?
There is absolutely no link between the imports and images, so it will stay. Everything is in.
But if you have the same resource with the same code, the image will still be there?
Yeah, absolutely. Yeah. And if you have a large stock of images, it can be the case.
Another question. Sorry. If you have a large stock of images, we can do that. You send them to us, and we can do that automatically.
Absolutely.
Another question. So if you change things on the site, can you then download your CSV file updated?
No, not for the moment, but it can be done. It can be something we can add. Indeed. Actually, at the beginning, the process, we thought that it will be nice to import and not edit online, even if it's possible, actually. So we thought that maybe the CSV file will always be updated. But if you need indeed something from the website because you have edited a few resources, we can make something.
It'd actually be better to download it rather than re-downloading the updated CSV. It would be better to re-download the updated Excel.
Yes. Yeah. I can make something to export data, and it could be in CSV or XLS. It's okay. Yeah.
Because that way, if you change something on the website, then you don't have to go back and change it on your original file. You just download the updated file. That'd be useful.
So is it something useful for you? Yeah.
I think if you can edit on the site, it makes sense to have that. Yeah.
Okay.
Did you have a question?
Okay, I can just show you.
One second.
Okay, sorry.
Well, it's about updating the website. What if I update my CSV file? So I have to replace the previous file always?
Yes.
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 Okay.
Yeah.
Is that?
Yes, I thought that would be automatic.
Not this one. This one is the manual one.
You thought it would be automatic in what sense?
Okay, if I have my CSV file, I change my CSV file, and automatically it will update the website.
Well, you have to go and upload it. You have to remove the old one and upload the new one.
Yeah. Yeah, I thought that this would not be necessary.
I mean, that's if you put in place the API with the automatic system, then you don't have to do all this. But you can't just do your CSV file on your computer and then it won't just automatically upload itself. You have to actually go and upload it.
Okay, but if we have an online database,
Yes.
okay, linked inside in our sites,
yes.
Linked to a CSV file internally,
yes.
It's possible to do this
update? Then if we put in place the API, then you don't need to do the manual.
Okay. Okay.
Then that will be automatic. Yes.
Yeah,
we present. You need to have a online database linked to your CV.
Yeah.
Not necessarily, actually. Because if you're working with an Excel file on a daily basis or FileMaker, I got this before, if you can just host a CSV file somewhere that I can just access, so you can just save this CSV file regularly and just put it on this URL, on this address, and I will just fetch the data from this address, and it's okay. So you don't need a SQL database for that.
Yeah, but that means that it's not completely automatic because then they would have to update their CSV file on their server.
Yes, indeed.
I don't know if you can do that automatically in File Sender, can you? For example. That was a question.
For me?
Yes.
No, I can't access an SQL database unless I have the credential to do this. And I'm not quite sure this is the right way to do this. That's why I think the IT in charge of the website can easily make an extraction and that could be automatic on its side, just to have this CSV file always accessible. And I can, from the Trace website, create a procedure to fetch this CSV file. But for any automatic import, I need to be in contact with the IT person. It's easy for the RCC because I am managing the website. Okay. Yes. I'm going to just to show you how to import a second time, just to see when we have less resource and updated ones and new ones, and you will see that everything will be taken care. So I have prepared a second file. So this one, okay. And on this, maybe it's not useful. Okay, can you see the second new CSV file? Yeah. Not yet. Okay. Okay. So this is the FCC data May 23 version 2. And on this one, I still have FCC 11 to 20, and they were already present on the first CSV file. But I have just added, two new lines here, 21 and 22. I have two new resources in my collection. And I have just made one change on the FCC 11. The country is now Spain and not France. Okay. So I have one line with an update. I have 10 lines that have been deleted from the CSV, and I have two new lines, okay? And I'm going to import this. So I go back to the Trace website, and from the import page, always, I edit the import. Okay, can you see the form? Not yet. Okay. So I remove my previous file and I choose the new one. Okay, FCC data May 23 version 2. I just change this and I click on save and import. It should be pretty quick in my side. Okay, so maybe you have seen the progress bar. Okay, do you have the green message? Okay, so we have different information in this message, and if it's green, you're okay. It's okay, everything is fine. If it's orange or red, this is like the ABS status. You have something, you have to contact the people in charge. Okay, so we have 10 resource items that have been cleaned, and I am going to show you this. This is the 10 lines that were absent from the CSV. We have 2 new resource items, that's okay, and 1 is updated. So it's okay, it's what I expected. I'm going to check this. I'm going back to the advanced search. I'm going to look at the resource provider, and I can see that now the scroll fake culture collection has only 12 resources and not 20 like before. Okay, so if I just filter this, I can find anything I want, and I have the right FCC 21, the new one, it's okay here. I have the 22 here. Okay. This is sorted by Genius, actually, okay. And just the last thing to show you is the FCC 11 here. And you can see now that this is in Spain, okay, not in France. And of course, the ABS status has changed too. Okay, and this is microRG. So I just wanted to show you that if your CSV file is updated and is quite modified, like this one, we have half resources less, and we have 2 new ones and a date. Everything is taken care by the system, and you can just add this CSV file again and again and again just to update everything on your collection. Last thing I need to show you is the cleaned resources. Is that okay with you? Yeah? Okay. So I will just log out to log as administrator, the super user. Okay. Just to show you that I have many more things to do when I am an administrator. Can you see the scroll page? Not yet? Yeah. When you see scroll, just say it. It's okay? No. Okay, so can you see the right page? No?
CSV page.
Oh. The CSV? Oh. Okay, great. So I would like to show you that you can see the Trace Manager Tools menu is far different from the previous one, because you can see here that we have all the import procedures for any culture collection that is already on the website. Okay, so I can just act on each one of them, but when you are logged in as a manager, you can just act on yours, and that's for safety, of course. Okay. And if I just go on content here, I will see the last updates of the website. Okay, can you see the table and the list? Yeah. And just to show you that the 10 lines that were removed from the CSV are still here. FCC 10, 9, 11, 7. But as you can see here, they are unpublished. So this is cleaned, so you can't see them. Users can see these kind of resources. But if you have made a mistake and you just tell me, oh, I need these lines back, I just have to publish it again, and you will just recover these 10 resources. Okay, so it's not deleted. That's a safety tool. Okay. The last thing I wanted to show, but maybe this is for this afternoon, this is the country's ABS compliance. Should I tell a word about this? Okay, good. So if I just go to the new page, country's ABS compliance, and this is, as Ian said, for Ian and Arnaud, but everyone can just view and browse this page, of course. But if you want to edit, this is only when you are logged in as a chief manager. And on this page, you can see also that some country has no information. So I have put this little sentence here, need information. And you just have to click on the edit button here. And from the form, you can just add the right address for the ABS clearinghouse. And here you can just choose among the different status and remarks. If we keep the remarks, because I have noted this morning that maybe it won't be necessary on the website, we'll see this. Anyway, you can just edit for each country the pre and post Nagoya Protocol status. And after that, the system will know what happened with this country pre and post. And depending on the sample date, it will show you the right ABS status. Okay. And here you can, of course, search here from country and just click FR and you just find the France, etc. I think it's good for me. Okay. Oh, yes, last thing, because I suppose that some people are wondering how to have an account. Yeah. How to get an account.
Yes, to get an account. Yes.
So I have just prepared this quickly this morning when I was listening. If you need an account to manage a collection, so you feel free to contact me, of course, no problem with this. But I will need some information to start with this new collection and import procedure. So I need the name of the manager, and you can have multiple people on this, but for the first time, maybe one is okay. So first name and last name, of course. An email, this is required to create an account on Drupal. The logo and the name of this culture collection. The university or institution which is hosting the collection. For example, for the RCC, this is the SBR. The website address, the homepage, if you have one. The specific pattern, you remember, for the Roscoff Culture Collection, we have this RCC strain details / and the RCC number for any strain on the website. If you have a specific pattern like this, you can provide me this beginning of address, and I will use it to automatically redirect users from Trace to your website. And finally, the first dataset. You don't have to give me anything you have directly, because of course, you have some work to do on this before. But if you have only 5 or 10 resources, you can just provide me an Excel or CSV file, and I will just start for you. Everything will be built, and it will be your turn to play with it later. Okay.
Why did we not put a request account on the website?
Because if you let anybody request an account, you will have a lot of demands. So I can just open it, but we thought at the beginning that it was nice to choose and to just contact people by email. But we can change this if you like.
We'll think about it.
Yeah.
Okay, so any questions? There's one thing I just wanted to show you actually as well, is the worms.
Oh yeah. I will just stop sharing my screen. Okay. And thank you for listening to me.
Board. One. Why? That's not where it lives. So the worms tool. Let me just go here. So the worms tool, if you click on that, it will go to this page, which is on the worms page. And basically, if I remember correctly, what you do is, so 1,500 rows at a time. You put your file, so it can be an Excel file just with the names of the species. You upload it here, and you do next, and then it will send you back basically a file with the names and all of the worms numbers. So very easy to do, and very useful. It means you don't have to go one by one. So. Okay, so any questions, comments? Happy with that?
Everyone needs a lunch.
Yeah, it's lunchtime. Yeah, and I guess you even more because it's
yeah, it's 1:30.
Well, it is lunchtime, so okay. So I think we'll maybe stop there for this morning. If you yeah, so just to remind you that we've showed you the upload procedure. If you don't want to do that, you just send us the Excel file and we'll take care of the upload ourselves. It's not very complicated, but we can do that. And if any of you have any, if you look at the site at any point, you have any comments about things which are missing, like the ABS status on the search and stuff, please do let us know, and we can modify. So the idea in the coming months, and so the EBB extension has to be finished by mid-July, if I remember correctly, which is kind of quite soon. So we're going to do a Zoom version of this for the other EMBRC people in the coming, I don't know exactly when, but before mid-July. So we'll get other EMBRC members to upload their data, and then the idea is that this will be integrated into the EMBRC website. So it will be virtually integrated. In any case, it won't be physically integrated, but this will be available on the EMBRC website, and then we'll see whether I guess we can track the users of this and stuff, and see whether it is something which which is useful, and the idea will also be to make some some publicity on that this tool exists, and hopefully it will be useful. Yes, yes. Yeah, and in fact, so yeah, I mean, from now on, the idea is that all EMBRC members can start putting their data in. I guess we will, from now on, probably, I don't know, I haven't talked to Fabien about that, but take off the EBB label and put on the EMBRC label. It's just some, some kind of changing of the logos and stuff. And it does also mean that we will remove data from, unfortunately, from the EBB partners who are not part of EMBRC, and the main one is the CCAP. I mean, I talked to Nick about this, and his opinion was that this is for EMBRC members, so it will be just the EMBRC. But yeah, from now on, we can start uploading and doing stuff. So yes. The question is, yeah, will it be maintained long term? Well, yes, I hope so. I mean, we already have a number of years of maintenance. I mean, to maintain this, there is a cost for maintenance of the website, which is not enormous, but there is an annual cost for that. And I would say with this kind of website, and I think EMBRC in the future would be willing to pay that cost. But the main point is, if this is something which is useful, on the one hand used by the users, and on the other hand, if it is something which is maintained, so this kind of resource, and I know from experience, during a project you upload your data and then you forget about it, and so it would have to be, particularly if it's manual uploads, we would count on the providers to actually do the updates, because if it doesn't get updated, then it will quite quickly, I think, become obsolescent. I think the main thing is if it is used. If it's used and the EMBRC sees that it's useful, I think that EMBRC will maintain it over the long term. Yep. Okay, so if there's not any more questions, if you do have any more questions in the coming days or whenever, just you can just contact myself or Fabien. And we're very happy to have any feedback on that. So let's go to lunch. So thanks, Fabien.
Thank you, everyone. Have a good lunch. And I will just go watch the YouTube channel all these days. So if you have questions, I can just maybe answer on the comments or something like this. Okay. De rien.
Bon appétit.
Bon appétit, mon ami.